Welcome to Pharmacognosy Laboratory. Today we will see experiment number 5 to study morphological and microscopical characters of cinnamon bark. So this is the sample of cinnamon bark. First we will study about morphological characters and then we will go for microscopic characters. Let us see what is cinnamon bark. The synonym of this drug is Ceylon cinnamon or Kalmi dalchini. This is a very familiar drug. You might have seen it in your house. It is used as a condiment. घरी तुम्ही सगळ्यांनी बघितलेलं असेल दालचिनी हा जो पदार्थ आहे तो आपल्या मसाल्यामध्ये वापरला जातो. So that is nothing but our cinnamon bark. The biological sources, it consists of dried inner bark of Cinnamomum zelanicum belonging to family Lauraceae. Now we will see morphological characters, color, order, taste, size, shape, etc. The color of cinnamon bark externally and internally differ. See, this is the external part and this is the internal part. Externally it is yellowish brown or grayish brown in color, while internally it is reddish brown in color. Then odor is slight or pleasant. Then taste is sweet and aromatic. The shape of bark is quill shape. Quill shape means what? When the bark is dried, the one end of bark gets shrinked inside and the shape which is obtained is known as quill shape. The size of bark is generally 1 meter in length, but these pieces are cut into small size pieces. So these are the different characters. Extra features are on external surface you can see the cracks, longitudinal cracks are present and there may be remains of the sting. So by these characters we have to identify the drug first and then we will go for microscopy. For microscopic characters we have to take the boiled crude drug, say like this. Then we have to take wash glass and take water in the wash glass. Then cut the crude drug with the help of a sharp blade and transfer the thin sections into wash glass containing water. Like this, you have to collect the thin sections. You have to cut 10 to 15 sections and from these sections we have to select the thin one. Then we will go for staining purpose. Now in another wash glass, take the thin section and stain it by using chloroglycinol and concentrated HCl. Put 1 drop of each. Then we have to wait for 5 minutes until the staining is done. In another wash glass, take the thin section and stain it by using iodine solution. Put 1 drop of iodine solution and wait for few minutes. Now staining is complete and I have prepared the slide by putting the section on slide, then put 1 to 2 drops of water and slowly keep the cover slip on it. Now adjust on the microscope to visualize the section. Now this is the diagram for TS of cinnamon bark. See, this is the upper cork layer having the rectangular cells. This is the cortex. Then here are the starch crystals present in the cortex cells. This part is known as scleroid. In cinnamon and cinchona bark, you can differentiate by this part. In cinchona bark, this part is not present. Otherwise, all characters are similar, but this part can be used to differentiate these two barks. These are the pericyclic fibers which are present in the scleroids. So when you stain the section by chloroglycinol and concentrated HCl, this part gets stained pink in color. Now This is the secondary phloem. Then acicular raphides. These are the strands of cells known as medullary rays. These are phloem fibers and oil cells. So when we stain the section, which part we have to visualize? This part. So let us see. Our staining is complete and we have adjusted the slide. Now see, this is a section of cinnamon bark. You can observe the pink color staining with fluoroglycinol and concentrated HCl, which is due to the presence of pericyclic fibers. They get stained. Other portion is not stained, you can see. But the pericyclic fiber part is only stained. Now this is the second section, stained with iodine. When we stain the section by using iodine solution, the starch crystals which are present in the cells, they get stained blue in color. So you can see the dark part, blackish blue part, which is due to staining of iodine. So in this way, we have studied the morphological and microscopical characters of cinnamon bark. Hope you have understood this. Thank you.